Okay, good evening, everyone. Thank you very much for joining us this evening for the Better Brains seminar. Before we begin, I'd just like to acknowledge the traditional owners of the land on which we meet today and pay our respects to their elders past, present, and emerging. So my name is Yen, and I'm the primary investigator of the Healthy Brain Project in the Better Brains trial. I'd like to welcome all of the attendees. I know we've actually been quite overwhelmed by the interest in this seminar. So I hope that you find it helpful and useful. I am proud to say that we have a stellar lineup of speakers tonight. So I do hope you enjoy the session. So this is the outline of tonight. We're going to spend a little bit of time talking about cognition, but primarily about we're going to focus on dementia prevention and risk reduction, and we'll also be speaking about how while a big part of dementia is genetic and is inherited, there is also a large part that is certainly modifiable. And our speakers are going to give us a little bit of insight into how cognition changes as we age, and what can be done to do that. So just a little bit of housekeeping before we start. If you have any questions, please write your questions into the Q&A box, and we will discuss them at the end of our session, in our Q&A session. So we have about 20 minutes. We were supposed to host this in person and on Zoom. Unfortunately, because of the winter surge in COVID cases in Melbourne, we've decided that it's the responsible thing, I think, for us to shift to an online only seminar. I hope that it does make me a little bit sad that we can't meet and gather in person, but hopefully we'll be able to do that safely in the future. So if you have any questions, please feel free to put it in the Q&A. If you don't, if you're uncomfortable with asking your question, you can also send it as a private chat to one of our co-hosts or our panelists, and we can address those questions for you in the Q&A session. Halfway through, we will have a bit of a break, so a 10-minute break, just to stretch our legs. Quick toilet break, quick drink of water before we proceed. If you need to drop off at any point, please feel free to do so. I realize that it is a school night, and we will be sending a copy of this recording to all attendees after the session. If you don't receive it, please email us at betterbrains@monash.edu, and we will make sure that you will receive a copy of it. Okay, we are actually just waiting for our first speaker, Paul.
Paul is here.
I'm here.
Oh, he is. Okay.
Yeah.
Okay, so I'd like to introduce you to Paul Maruff. Professor Paul Maruff is a neuropsychologist with expertise in the identification and measurement of subtle behavioral and thinking and memory dysfunction as people get older. Paul's research integrates conventional and computerized neuropsychological testing with cognitive neuroscientific methods to guide decision making in drug development and in clinical medicine. So tonight, Paul will be talking to us a little bit about the changes in cognition we can expect as we age. Paul?
Thank you, Yen. Thank you, everybody. I noticed apropos this talk that the photo of me on this is much younger than the head you see talking. So it's very relevant. So as Yen said, I'm going to just seek to set the scene by trying to outline some of the philosophical basis of what we're doing. So as we in the community sort of often believe that as we get older, as people get older, there is a consequent loss of their ability to think effectively and act purposively. If we look at a word cloud from articles about thinking in older people, you can see that the words, the adjectives that come up quite a lot are generally negative. and this is consistent with medical models of how we often believe and think about aging and cognition. Thanks, Yen. Can I drive these or?
Okay. I'm driving them.
Driving. Okay. So from the scientific side of things, if you look at the figure in the middle of the screen, you can see on the bottom it says chronological age. So that's age in years. And on the y-axis, you can see a score. And largely you can see that the majority of the lines moving from left to right are moving in a negative direction, essentially showing evidence from large groups of people who have done assessments of different aspects of their thinking, that as you move from 50 through 60, through 70 years, through 80 years, there's a pretty linear and irreversible or decline in cognition. One aspect does seem to improve, which they call vocabulary, which seems to improve as you get older, although it really just stays the same from when you're 50 through 80. But all of these other aspects of thinking, like your ability to remember, your ability to solve problems, your ability to solve puzzles, seems to deteriorate. And this model is so pervasive that in the current textbook of gerontology, this is given as the cornerstone of how gerontologists are trained to actually think about the way in which they should consider that thinking changes in older people. Thanks, Yen. One of the principles that you'll hear about a lot tonight is that if we take dementia out of the picture here, we can show that there's a lot of aspects of behavior and experience in midlife that can actually have some relationship to that deteriorating change in cognition, or that deterioration in thinking that we have mapped over the past couple of years. And whilst there are a number of different factors that have been shown to predict that, these ones that are shown on the figure here seem to have some consistent and reliable relationship that really does occur often. And importantly, what I've done is divided these into two groups. The top, age, sex, and carriage of a gene, we can do nothing about. But the ones that are in the lower clump, if you like, education, blood pressure, body mass index, cholesterol, and physical activity are all what we call modifiable risk factors. Thanks, Yen. Interestingly, too, when you think about aging, trying to understand it is a moving target. If you look at the date of the publication of this paper, 2004, and if we say that at least took 4 years for it to get finished or into press, then the 80-year-olds in this represented on this data were actually in their 50s in 1970, and the 70-year-olds are in their 50s in 1980. So these older thinking reflects a midlife that was, 80 years ago ourselves now. So if we move on and think about that, about what aging is in the 21st century, thanks, Yen. You can see that since the turn of the century, we've had a growth in widespread statin use, so cholesterol levels have come down. The National Tobacco Campaign has diminished or reduced smoking levels. Blood pressure, widespread drug pressure for people with low cardiac risk happened in 2003. All of the dietary recommendations for lowering salt and increasing vegetables for blood pressure occurred at the end of the 20th century, and aggressive and assertive management of diabetes and pre-diabetes also became a big deal. So all of those people who you see in that figure had no benefit of those changes in midlife. The people who are coming into our studies now, such as people of my age, have all had the benefit of turning through their midlife in a circumstance where our general health from our cardiovascular health is relatively well looked after. Thanks, Yen. If we take out another thing, as we understand now, that there's brain disease, which is not part of normal aging, but our work has shown that actually brain disease or changes in brain biology that cause or related to the appearance of dementia actually begin many, many years before someone becomes so unwell that they're unable to look after themselves. And a number of groups around the world, including us, have identified that there's specific protein fragments that can be detected in the brains of older people. And if we follow those people over time, we see that their thinking gets progressively worse, even if they don't meet any clinical criteria for dementia or even a relatively lax criteria. So the figure at the bottom on the left-hand side shows in the just the change, a deterioration in thinking that occurs in individuals who are carrying these protein fragments. And you see that individuals of the same age, same gender, same education, who don't have this, what we call amyloid, you can see that there's no deterioration in their cognition at all. And it's important too to look at the time frame over which that is being measured. That's 8 years. Right, so 60 to 70-year-olds. So we saw in those figures I showed you before quite substantial decline from 70 to 80, from 60 to 70. But here we're showing data in people who are not declining at all if you remove those folks who have got the occult disease, so the hidden brain disease. That is, the majority of people don't have that. So at worst, it's 1 in 4. But that means 75% of older people don't carry that sort of disease. And this is what we're interested in. Thanks, Yen. And if we then look at groups of people where we take out brain disease and we hold constant, or rather we select them to be older in the 21st century, so they've had all of those benefits to midlife cardiovascular health, and we control then the extent to which they have brain disease. I'll just flick you through some figures now. So Yen, the next one, please. So what you're looking at here is the red data is a complete sample. And then the blue data, the data shown by the blue line, is if we just restricted the analysis to those people who did not have any brain disease. So the red line reflects the combination of brain disease and normal aging in people who are not demented. And then the blue sample is the same people, but with the brain disease removed. So you can see that if you analyze data from older people and you don't know what's happening in their brain, so you don't understand the level of brain disease, you may make an incorrect assumption that thinking gets worse as people get older. If you actually just study cognitive aging, you see that there's no decline at all. Thanks, Jen. And that's for remembering. This is for short-term memory. You see exactly the same pattern. Thanks, Yen. You can see that rapid decision-making, the same thing. A complete group of people, older, non-demented, but followed for a long time, show decline. Those folks who have got no brain disease show no decline. And then finally, if we look at the speed with which people make reactions, you can see that that deterioration happens irrespective of whether you've got the occult brain disease, so the amyloid hidden in their brain, or whether you haven't, suggesting that the speed with which we act slows down as we age. Thanks, Yen. So my starting point here is I want to say provocatively that I don't believe, or some of us don't believe, that there's any such thing as cognitive aging. Once you control the effects of brain disease, aging is not really associated with a loss of quality of cognition. It is the case that brain disease becomes more common with age, but many of the factors that related to brain disease we can actually control. And those are by doing things that look to control those aspects that we showed in midlife and about which you're going to hear much more of. And the most important thing is that the earlier we begin to control and take care of those factors, the better it is. So thank you very much. I'll stop there.
Thank you so much, Paul. So again, if anyone has any questions, please pop your questions in the Q&A box and we will get to it at the end of our speakers. So our next speaker is Professor Amy Brotman. She is a stroke and cognitive neurologist at the Royal Melbourne Hospital, Austin Health and Eastern Health, and a clinician researcher at the Victorian Heart Institute at Monash University, and was previously also based at the Florey Institute. Her research focuses on the links between stroke, vascular risk factors, and neurodegeneration, with an interest in imaging brain network degenerations following stroke and the diagnosis and management of focal onset dementia. So today, Amy will be talking about the steps we can take to have a healthier brain and body. Amy?
Thanks so much, Jen. Thanks, Paul, for that introduction. That's great. So I have prepared a series of slides for you. So it's going to be skates on for poor Yen, because I'm going to whiz through some of these data and also talk a lot about what you can do. I know there's also lots of questions which we can get to at the end of this presentation. Next slide, please, Yen. As you've just heard Paul allude to then, I think that our models of healthy brain aging are really changing, but also our understanding of what causes dementia, all-cause dementia. We were taught that it was something that was inevitable, which we think now is just not true. But the bad news is many lifestyle factors do cause accelerated brain aging, and I'll kind of run through those with you in a moment. And essentially almost all of those factors are vascular risk factors, hence my interest in stroke and in dementia. We know that vascular risk factors cause stroke and heart disease, that's very accepted, but we also know that stroke and dementia are absolutely linked. But the good news is you can do something about it. And we do know that that those are most at risk actually get the most benefit from neuroprotective behaviours, which is the term that I like to use. They're the ones that protect your brain. Next slide, please, Jen. So we know that the risk factors for late onset dementia, and I'll just make that distinction because we divide dementia into early and late, late being over the age of 65, early being less than 65. The people who develop dementia who are less than 65 often have a history of traumatic brain injury globally, is the commonest risk. And in high income countries, these are often genetic dementias as well. But the risk factors for late onset dementia are exactly the same as those for stroke and heart disease. High blood pressure, smoking, lack of physical activity, which I'll speak more about in a moment, adult onset type 2 diabetes, not type 1, obesity, having a high BMI, and which is body mass index, early life lack of education, hearing loss is a strong risk factor that is poorly recognised, and I'm going to talk to you all about getting your hearing checked because nobody has deafness apparently, according to my patients. Depression, social isolation, and as Paul said, your carriage of a particular gene, which in all of the studies where we look at genetic risk comes out as the #1 factor. So that's something you can't modify, nor can you modify how much education you got as a child, but you can modify almost all of those, the rest of them. Next slide, please, Jan. So the risk factors for stroke are the same. In fact, ApoE4, when I was in medical school, was in fact taught to us not as a risk factor for dementia, but as a risk factor for cardiovascular disease and stroke. Raised blood lipids are probably more of a risk factor for stroke and heart disease, particularly for heart disease, than they are for dementia, and also atrial fibrillation, which is an irregular heart rhythm. But we now know that atrial fibrillation is also associated with dementia. So they're the same. Next slide. When we look at what happens to our brains when people have vascular risk factors, when they actually measure how much the brain shrinks over age, we've seen associations with all of these vascular risk factors. So I talked at the start, I used this term accelerated brain aging. One of the critical things about brain aging is shrinkage. It's normal for our brains to shrink. Just as our skin shrinks, we get wrinklier, we lose height because our bones and the discs between our bones lose volume. That's actually a normal part of aging. So that brain shrinkage doesn't mean you're losing brain cells. However, pathological brain aging can mean that you are actually losing brain cells. And again, when they've done imaging on people with these risk factors, certainly midlife and untreated hypertension, midlife type 2 diabetes, midlife obesity, smoking, low levels of physical activity, etc., are associated with greater brain shrinking. So you can actually cause your brain to shrink more if you're having these bad brain behaviors. And the good news is you can do something about that. Next slide, please, Ian. So this is what I've often given these public talks, talking about the risks between stroke and dementia and vascular risk factors, and literally the audience looks like this to me. Everyone's looking absolutely horrified, and the arms all go up at the end of the lecture. What can I do to prevent this from happening to me? Next slide. So the two things I say are get checked and get engaged. I don't mean engaged to be married, but you can if you want to, because social isolation is something that is bad for your brain. I'll talk about what I mean by getting engaged in a moment. Next slide. So if I go back to that slide and talk about what we can do to prevent accelerated brain aging, pathological brain aging, we'll flip it around and we'll look at, in fact, what things have been proven to be protective for our brain. This is from a Lancet Neurology piece that was published some years ago now, but as you can see, it's the reverse. It's the inverse of all of what I said before. Stop smoking, start exercising, adhere to the Mediterranean diet. And we know that all of these things reduce inflammation in the body. We actually talk about a low inflammatory diet now. We know that exercise has profound effects. It is a panacea for brain health, it really is. We need to talk about increasing our social network. Next slide, please. So get checked. I always say heart health is brain health. In fact, I tweet about it. So get your vision checked, because we know that if you can't see, then you're getting less stimulation. You're more likely to be socially isolated. But people who are blind from birth don't have an increased risk of dementia because they get out there and they stay social and they interact. Get your hearing checked. I cannot tell you how many people say, oh, there's nothing wrong with my hearing, when I'm screaming at them in the room. The fact is, we know now that low hearing or hearing loss is definitely associated with late life dementia. So get your hearing checked. If one of your relatives has said to you, gosh, my hearing's not great, or gosh, you know, grandpa, can you please get your hearing checked? Maybe listen to them, ironically. Get your blood pressure checked, get your blood sugars checked, get your blood fats checked. Just making sure that you don't have high blood pressure, diabetes and cholesterol. Get your weight checked and try to be as much as possible within your body mass index. It's hard, but it is something that's really important. The COVID years have also been associated with COVID kilos. So try to do something about those. And you'll hear later, Shant, that we'll be talking much more about this. Get your sleep checked. Some people who snore might actually have sleep apnea, and this can be associated with significant brain aging and stroke. So think about getting a sleep study if you snore or if you are very sleepy during the day. Next slide, please. So get engaged is actually social, mental, and physical engagement. Next slide. So social and physical activity in dementia are very, very important. When we talk about physical activity, we know that not only is midlife physical activity important, but so is late life. So we have to be active and stay active. We know that low levels of midlife physical activity, when they stay low, are associated with a higher risk of dementia. And this has been seen in now thousands of people. We know that higher midlife fitness levels, and remember, midlife starts at 35, just saying, don't judge, as my daughters would say, is associated with lower hazards of developing all-cause dementia. We know that maintaining physical activity in late in life reduces your dementia risk and slows your dementia progression. But you also need to stay socially active too. All of the studies have repeatedly shown that dancing is associated with the lowest risk of dementia. That's probably because it's that combination of both social activity as well as being physically active. So when I'm saying get engaged, Don't necessarily do a Sudoku on your own or do a CrossFit on your own, which are great, but get out there, do something, learn a language, join a walking group, volunteer, visit a friend, take your grandchild to the park, and you get on the swings. Next slide, please. So to summarize, vascular risk factors are also risk factors for dementia. And we know that stroke and dementia are linked. So I think these data are now irrefutable. There's certainly evidence that dementia might be preventable in a large number of people. Up to 70% of dementia risk in some studies is modifiable. So if we get checked, and if we get lean, and if we get fit, and if we get social. And intervening with exercise might stop your brain from shrinking. So this is something that we're looking at now. Next slide. A lot of my patients will say to me, oh goodness, I'm too old, or I can't do it, or I can't maintain my exercise as I get older. So here are two people to inspire you, not me, with my Swami in Kerala some years ago. This is Nanamal, who teaches 100 students. She can still do a headstand. She can more than touch her toes. And this is Tao, who's been a star of a 2017 Athleta advertising campaign. Next slide. The thing these people have in common is they're both 98. So I think that this shows that we can stay active, we can stay engaged, we can stay fit until very late in life. It's worth the effort. And here are my two marathon runners that I still follow, one aged 82 and one aged 91. And Ed did a sub 4-hour marathon aged 82, which is just phenomenal. So all of you, next slide, please, Yen. Move, stand up, move. Thanks very much.
Thank you, Amy. Those are pretty incredible people. Oh my gosh, sub 4-hour marathon.
I couldn't do it sub
82. Thank you very much. Okay, so our next speaker is Lisa Bransby. She is a final year PhD candidate at the Turner Institute for Brain and Mental Health at Monash University and a recipient of a Dementia Australia Research Foundation PhD scholarship. Her research focuses on modifiable risk factors for cognitive decline and Alzheimer's disease dementia in middle-aged and older adults. And Lisa will be sharing some of the data that she collected and analyzed as part of her PhD today.
Thanks, Yen. Hi, everyone. I'm really excited to be here with you all today and share some work from my PhD on modifiable dementia risk factors in middle-aged adults. Thanks, Yen. So as many of us here would know, dementia affects so many people and is the leading cause of disability and mortality worldwide. It is a clinical syndrome characterized by impairment in memory and thinking abilities and dysfunction in daily life activities. Alzheimer's disease is the most common cause, and unfortunately, we still do not have a cure or effective and accessible disease-modifying treatment. And because of this, it is really important that we identify and understand alternative ways in which we might be able to combat dementia. Next slide, thanks. So this figure here illustrates the most dominant hypothesis for the Alzheimer's disease trajectory. I don't have time to go through it in great detail today, but the main message that I want you to get from this figure is that Alzheimer's dementia progresses extremely slowly. For some people, it can take decades. And this is important because it may mean that particularly earlier on, there may be a window in which we might be able to intervene and stop Alzheimer's disease in its tracks. And this may be possible potentially when individuals remain cognitively normal, meaning they have no brain impairment in their memory and thinking abilities, but they have subtle and insidious pathological changes that are already occurring in the brain. So this is not going to be easy. There are big questions that need to be answered, such as what makes someone at greater risk of developing Alzheimer's disease and eventually the dementia syndrome? Can we identify these people? And if we can we even intervene much earlier than that? How do we identify these people and when, and is there anything we can do about it? Next slide. As you've heard from the previous speakers, we know that increasing age is the greatest risk factor for dementia. There are also genetic risk factors, with the ApoE4 gene posing the strongest risk for Alzheimer's dementia. And sex may also be a risk factor, with more women developing Alzheimer's dementia than men. And as these risk factors really help us in being able to identify who is at risk of Alzheimer's dementia, and also help us understand the natural progression of this disease. But in terms of approaching dementia intervention and prevention, there really isn't much we can do about these risk factors. We can't modify them. Thanks, Yen. As you've already heard, there are many dementia risk factors that are modifiable. This figure that you saw on Amy's slides as well is from the 2020 Lancet Commission of Dementia Intervention, Prevention and Care, where they identified 12 modifiable risk factors for dementia that have the most robust evidence. There are also other risk factors that have been identified as well that are also modifiable. And it's important to note that these modifiable risk factors relate differently to dementia risk at different life stages and across different groups and populations. So for example, as we've had before, low education is a risk factor for dementia when present in early life. So these 12 risk factors were estimated to account for approximately 40% of dementia cases, which means that if these risk factors were reduced or eliminated, some dementia cases may be preventable. Thanks, Yen. When we study and we try to understand these modifiable dementia risk factors, it's really also really important to consider how modifiable they are. Is it feasible to target these risk factors at multiple different levels to reduce dementia risk? So, for example, air pollution, as we know, is a risk factor for dementia, and it is modifiable, but can we tell an individual to change the air pollution where they live, or to move to a less polluted area in order to reduce their dementia risk? Rather, this is a risk factor that would need to be targeted on a population-based level, either through government policy or large-scale societal change. But it is much more feasible for, to advise an individual to reduce their smoking or increase their physical activity to reduce their dementia risk. So the risk factors like these are readily modifiable by the individual themselves, with or without professional guidance or intervention, and that would really depend on the risk factor, the person, and the circumstances. So these risk factors at the bottom level of this figure are the ones that I focus on in my research. Thanks, Yen. It's also really important to consider that many of these modifiable dementia risk factors are related to each other. Some, if not many people, have more than just one at a time. And these images here are just to provide a bit of an example. So let's say you have some junk food, you eat KFC for lunch, you may not feel up to going to the gym or doing some exercise that day. So you may just sit in front of the TV instead, because you haven't gone and you're exercising, you may not sleep as well. And then because of this, your mood may be affected, so you may feel a bit down or a bit anxious the next day. And because of that, you may opt for the less healthy food option because it's a bit more comforting. And this scenario can go on and on. These arrows are double-ended because this could go in the other direction. So the main message here is that it's important to consider that these modifiable dementia risk factors may not act independently of each other. And it's important to be able to investigate combinations of these risk factors and how they relate to cognition and dementia, as this could hopefully provide a more accurate estimation. Thanks, Yen. So for the research that I will share with you today, I work with data from the Healthy Brain Project. Many of you would know it, some of you may even be participants. So this is an online study that seeks to understand midlife risk factors for dementia. Participants are aged 40 to 70 and are cognitively normal, meaning they have no cognitive or memory and thinking impairment at baseline or when they enter the study. Participants are residents of Australia and fluent in English. So participants sign up online. They do a bunch of health and lifestyle questionnaires, some memory and thinking tests, and also provide a saliva sample for genetic sampling via a kit that we send them in the mail. Thanks, Yen. For this research, we selected to include dementia risk factors that are readily modifiable at the individual level. And you can see the risk factors that we included on the left side of this figure. Because as you can see, there are quite a lot of them. We just wanted to synthesize them in a meaningful way. So we classified them according to the 5 broad domains that you can see on the right side of this figure. And these include mood symptoms, lifestyle behaviors, cardiovascular conditions, cognitive engagement or mentally stimulating activities, and social engagement, and lastly, sleep disorders and symptoms. Thanks, Jen. So from the Healthy Brain Project, we took a sample of participants of about 1,600 people, and we wanted to determine the prevalence and the overlap between these 5 domains of modifiable dementia risk factors. So this figure shows the percentage of participants that reported at least one modifiable dementia risk factor in each of these 5 domains. So what we observed was that 93% of this sample reported at least one modifiable dementia risk factor, meaning that almost everybody had at least some modifiable dementia risk. And 67% of participants had at least one risk factor in 2 or more domains, meaning that, again, the majority of participants had risk across multiple of these domains. Thanks, Jen. Okay, so now we know that most participants in our sample had multi-domain modifiable dementia risk factors. How does that relate to their cognition? So we looked at differences between groups of participants that reported 0 modifiable risk factors versus those that reported risk factors in 1 to 5 of the domains. And we looked at the differences between these groups on 3 different outcomes of cognition, which you can see across the bottom of the figure there. And these include attention, memory, and subjective cognition, which is subjective concerns or worries about one's own cognition or memory and thinking. Across the vertical axis of this figure, you can see the effect size value, meaning how big is the difference between the groups. And on the some of the bars in the figure, you can see some little asterisks, and that means that those differences were what we call statistically significant or statistically meaningful differences. So what we saw was that individuals that reported these modifiable dementia risk factors in 3 to 5 domains had worse memory performance and greater subjective concerns about their own cognition. And based on the effect size value, these differences range from about moderate to large. And also individuals that reported risk factors across all 5 domains also had worse attention performance. Thanks, Jen. So what does this all mean? So we, in a group of middle-aged adults, we observed that modifiable dementia risk factors across multiple domains of life are highly prevalent and associated with worse cognition. So this could mean that having multiple modifiable dementia risk factors, particularly across different areas of life, may be associated with early and subtle negative neurological changes, and that dementia risk reduction in midlife may be necessary. It's also important to consider how readily modifiable these dementia risk factors are. Is it feasible to target in intervention trials like BetterBrains that you'll hear about a bit later? And how easy is it for individuals to make these changes and sustain these changes in their everyday lives? It's also important to acknowledge that there are many barriers to dementia risk reduction, even if the risk factors are readily modifiable, such as access to healthcare, education, socioeconomic status, as well as the fact that humans are not always great at changing their behavior and maintaining that change. So these are things that we definitely plan to explore and address in the future. This research supports that modifiable dementia risk is multidimensional. People have risk across multiple areas of their lives. So it's really important to try to understand the impact of a wide and diverse range of risk factors. And finally, studies like BetterBrains will be really important in telling us whether targeting these multi-domain modifiable dementia risk factors in midlife will maintain cognition and prevent future decline. Thanks, Jen. Just like to very, very quickly acknowledge my mentors and the investigators of the Healthy Brain Project, my research team, the BetterBrains team, especially for all their hard work putting this event together, and most importantly to the Healthy Brain Project participants for the incredible contribution to this research. Thank you so much.
Thank you, Lisa. All right, we've heard 3 excellent talks already. I think just having a really quick look at the Q&A panel, you have a lot of questions, which a lot of which we'll get to at the end. There are a couple of questions about how we define midlife. And given that we've talked about midlife a lot, typically it's not well defined. You know, you'll find varying kind of definitions, but typically the period of life that we're talking about range from 40 to 65 years old, sometimes 35 to 65 years old. And that's what we consider a middle-aged or midlife period. So we're going to take a quick 10-minute break. So if everyone can come back again at 6:50, please grab yourself a cup of tea, you know, go to the toilet, and we will resume really, really soon.
Ian, do you want us to stay just on, just step away if you like?
Sure. Yeah, that would be great. Thank you.
Yeah, can I move the slides myself?
Yes, I will hand it across to you.
And so it'll be your presentation that I can then just take control
Oh, I don't think I know how to do that. Oh, no, I can do that with slide control privileges.
of. Can you just give me the slide control privileges and let me
Yep.
see?
I've just done that. Can you just check? There you go.
Brilliant.
Amazing.
Yen,
Technology.
It's Emily as well. Could you also give me
Sure.
slide privileges? Thank you.
Thanks, Yen.
Okay, welcome back, everyone. We'll let everyone trickle back in. I hope you managed to grab yourself a cup of tea in the meantime. All right, for those who joined us a little bit late, my name is Yen. I'm an associate professor at the Turner Institute at Monash University. And tonight we are talking about dementia prevention and risk reduction. So we've already had the opportunity to hear from 3 excellent speakers. And I would like to kick off the next session. And introduce you to Professor Shantha Rajaratnam. He is the Deputy Head of the Monash School of Psychological Sciences and Deputy Director of the Turner Institute at Monash University. He is an expert in sleep and circadian rhythms, including this role of internal biological clock in the regulation of the sleep-wake cycle and how clock disruption leads to sleep disorders and other physiological consequences. So today, Shantha will be talking about the association between sleep dysfunction or disruption with cognitive decline and dementia. So Shantha, off you go.
Thank you, Yen, and thank you to the BetterBrains team for putting this great session together. So I'd like to talk about one of the factors that some of the previous speakers also alluded to, and that is sleep and sleep health as a modifiable risk factor for dementia. So a quote from a famous sleep researcher is that if sleep doesn't serve some vital function, it's the biggest mistake evolution ever made, because it is a function that is highly conserved. And if we live to the age of 90, we would spend about 30 years of our lives asleep. So the assumption is that it must serve a very important function. Now, we've started to think about sleep health as one of the key pillars of health, and this is described in the context of mental health, but we could also replace that with cognitive health and think about some of the risk factors that we've been talking about so far. So I want to start by just giving you a brief introduction to how we think the sleep and wake cycle is regulated and what the kind of common models that we use to describe this. So we think the sleep-wake cycle is controlled by at least 2 processes. The first process is called the circadian process, and that refers to the body clock or the brain clock, and I'm going to talk a little bit more about that in a moment. The second is called a homeostat. The homeostat refers to a process that tracks our immediate sleep and wake history. Now, these 2 processes interact to create a cycle of sleep and wakefulness, and also a cycle of sleepiness to reach a point in time where we feel so sleepy that we can easily go off to sleep and have a consolidated sleep bout. Now, we know that the light and dark cycle is critical for regulating the circadian clock. And it's gated in humans by the sleep and wake cycle, because of course, when we're asleep, we don't receive light input. And as humans, we are terribly social creatures. And of course, our sleep-wake cycle is largely also determined by or impacted by our social constraints. How does sleep change as we age? Well, as a function of aging, and this shows an example of how different stages of sleep change as we age from 5 to 85. And so the first thing you'd note is that there is a reduction in the total amount of sleep. And then if we look at the individual stages of sleep, wake after sleep onset is a measure of how well consolidated our sleep is, how much wakefulness that's occurring after we fall asleep. REM is rapid eye movement sleep. SWS is slow wave sleep, the deeper stages of slow wave sleep, sometimes called delta sleep. And stage 1 and stage 2 are the lighter stages of non-rapid eye movement sleep. Now, you can see that the stages of sleep also change as we age, the composition of the changes of sleep. By the time we're 85, we see much more wake after sleep onset. We see a marked reduction in slow wave sleep, and we see some reductions also in rapid eye movement sleep. Now, this is important because sleep research has demonstrated that particularly slow wave sleep, but also non-rapid eye movement sleep, play a critical role in memory consolidation and new learning. So as our slow wave sleep declines as we age, this is potentially one of the mechanisms that explains why it's difficult for us to learn new things, to consolidate memories, and so on. Now, I want to spend just a minute talking about the circadian system because, as Yen said, it's a topic close to my heart as well. So we now understand quite a bit about the way that the circadian clock functions. We now know that as well as a circadian clock located in the brain, in the hypothalamus, we know that there are clocks distributed throughout the body in virtually every organ of the body. In fact, every cell has the capacity to keep this 24-hour cycle. We know that disruption of these clock processes expose us to a number of risks, and all of these are listed on the right-hand side. And you can see that impaired cognition is the first one that's mentioned because it's the most immediate consequence of disruption of these circadian processes within the body. Now, Claire Anderson and Jesse Manusakis did a study a few years ago looking at how, as we age, you may have heard that your circadian clock moves earlier in time. And so what they documented was that earlier circadian timing, measured by, in this case, the rhythm of secretion of a hormone called melatonin, was associated with increased memory impairments and poorer recall as well in a group of older adults who were reporting subjective memory complaints. So this suggests that the circadian clock and its timing plays a critical role as a risk factor in cognitive decline and memory impairment. What about how much you sleep? Well, there's been a number of studies, including this one recently, which was published in Nature Communications. And they showed that at the age of 50 and 60, if you sleep for less than 6 hours compared to sleeping for at least 7 hours, your risk of dementia is increased. And if you persistently sleep for less than 6 hours between the ages of 50 and 70, your risk of dementia is about 30% increased after controlling for other risk factors. And the little graph on the right-hand side is a sub-study this group of investigators undertook in which they looked at the risk of dementia on the y-axis here, and the x-axis shows the objectively documented total amount of sleep. And you can see the risk of dementia increasing as the total amount of sleep decreases. Now, what about manipulating the circadian system? Well, we know that light is the major time cue for the internal circadian clock in the brain. We know that light input is received and transmitted through a dedicated photoreceptor pathway, where the circadian system is maximally sensitive to short wavelength light in the blue range. And that's why a lot of products have come onto the market that are enriched in short wavelengths, because the circadian clock is maximally sensitive to light in this wavelength range. And we also know that there are now a number of ways in which we can treat sleep disturbances in individuals with dementia using light. And so this was a study that demonstrated that increasing intensities of light can improve the sleep-wake cycle and also reduce cognitive decline in patients with dementia. Now, What about sleep disorders? And Amy talked about obstructive sleep apnea. There are many types of sleep disorder, and obstructive sleep apnea and insomnia are perhaps the most common. And you can look at the distribution or the approximate prevalence of these disorders, and also RLS, restless leg syndrome, in across the lifespan. And you can see the increased prevalence in obstructive sleep apnea after we get to approximately the age of 45. Now, there are obstructive sleep apnea is characterized by brief pauses in breathing or breathing becoming shallower during sleep, which causes what we call intermittent hypoxia, reductions in blood oxygen levels, as well as fragmentation of sleep. Insomnia refers to difficulty going to sleep, staying asleep, or getting back to sleep after you wake up in the night. So let's just look at some of those factors and how they may impact upon dementia risk. So we know the mechanisms have been described in a number of studies linking sleep as a risk factor to Alzheimer's disease and vascular dementia, for example. We know that obstructive sleep apnea, as I mentioned, causes intermittent hypoxia, this decrease in oxygen levels in the blood, as well as sleep disruption. These result in processes like oxidative stress and inflammatory immune responses, which all contribute to increased beta amyloid levels, phosphorylated tau production, decreased glymphatic clearance, which is a pathway that clears waste products from the brain. And all of these lead to increased risk of dementia and both Alzheimer's disease and vascular dementia. Now, because the messaging here is that these are modifiable risk factors, indeed, all these sleep disorders are readily treatable. Obstructive sleep apnea, one of the most common treatments for obstructive sleep apnea, is continuous positive airway pressure, CPAP. This was a study that demonstrated in a cohort of looking at medical records from Medicare beneficiaries in the United States, looking at how treatment adherence to CPAP in obstructive sleep apnea patients reduces the risk of a diagnosis of Alzheimer's disease, other types of dementia, or mild cognitive disorder. Finally, I've covered a lot of information about sleep, and there is a lot of information available at the Sleep Health Foundation website. You can look at the website that we have over 90 fact sheets now in a range of topics, including aging and sleep and dementia and sleep, and you're welcome to look at that website to get more information. And finally, if you think that you may be experiencing a sleep problem, and as I said, there are many treatments available, you can contact your GP, who can give you a referral to a sleep specialist, and we have a sleep specialist service at the Turner Institute that focuses on body clock disorders, insomnia, and obstructive sleep apnea. So there are the contact details, and you're welcome to make contact if you'd like to see a clinician about your sleep problems. Thank you.
Thank you very much, Shantha. Okay, now just conscious of time, so I'm going to move straight on to Dr. Emily Rosenich. Dr. Emily Rosenich is a postdoctoral research fellow at the Turner Institute at Monash University. Her research investigates vascular, lifestyle, and genetic contributors to cognitive decline and dementia, and she's also very actively involved in the facilitation and data analysis of the BetterBrains trial. So Emily will be talking about how genetics and heart health might have different effects on thinking and memory decline, as well as brain atrophy as we age. Emily.
Thank you very much. It is such a pleasure to be talking through this work that we're doing here at the Turner Institute tonight. So I think I'm kind of preaching to the choir at this point of the seminar tonight. However, in recent decades, the incidence of both all-cause dementia as well as Alzheimer's disease dementia has actually fallen in many Western developed countries. And as Paul said at the very beginning of this seminar, we don't know exactly why this is. However, it has been hypothesized that this is likely due to improvements that have been made in people's access to education, healthcare, nutrition, as well as changes at the population level that relate to people quitting smoking, drinking less alcohol, and engaging in more physical activity. And interestingly, alongside a falling incidence of dementia, which is a reduced number of new people who are diagnosed with the condition, the incidence of cardiovascular disease and stroke has actually fallen in parallel, which is something that Amy touched on earlier. So this is data from a recent report by Frank Walters and colleagues, and they sought to determine the incidence of dementia over about a 25-year time span. And they reported that the incidence of all-cause dementia and Alzheimer's disease dementia has actually reduced by about 13% and 16% respectively per calendar decade over the past 25 years. And so this data actually got a lot of people excited because we currently don't have a cure for Alzheimer's disease. We have no effective or accessible disease modifying therapies. So a reduction in incidence might mean that at least some risk for dementia is actually modifiable. So I wanted to include this figure here, which has been talked a lot about tonight, which is from the Lancet Commission. And this paper covered 12 modifiable risk factors for dementia. But something that's quite striking about this figure or this table is that at least 6 out of the 12 risk factors reviewed actually relate to heart health, or at the very least have some type of implication for our heart's health. So there has been recent work over past decades that's tried to clarify associations between heart health risk factors and cognitive decline and dementia. Unfortunately, though, as we have said, not all risk for dementia is actually modifiable. So there's been multiple non-modifiable risk factors identified for the development of cognitive decline and Alzheimer's disease, these being of an older age, being of the female sex, as well as genetics. And I should make it really clear here that researchers haven't actually found any genes for late onset or sporadic Alzheimer's disease that are causative of the condition. All we have are risk genes. So ApoE4, which has been mentioned earlier today in the presentation, a carriage of at least one E4 allele is considered the strongest genetic risk factor for late onset or sporadic Alzheimer's disease. And so this is the one that I'll focus on in this talk with you tonight. So in light of the fact that there seems to be multiple modifiable as well as non-modifiable risk factors, the field has tried to develop what we call dementia risk scores. And obviously, as the name suggests, these scores try to predict or estimate a person's future risk of developing the condition. So these risk scores are really important to have because we need to be able to identify people who are at the highest risk of the disease so that we're able to then enroll them and stratify them into either drug trials or behavioural intervention trials to see if we can prevent the disease from happening. So a very common dementia risk score is the CADI risk score or the CADE. However, there are multiple other risk scores now, including the ANU-AD risk index, as well as the Lifestyle for Brain Health Index. And these composite risk scores capture multiple sources of information. So we ask people about their age, their sex, their educational level, their body mass index, physical activity, blood pressure, cholesterol, as well as genetic risk. And the CADI risk score in particular has become quite important in decision-making about risk and management of risk factors, particularly in older populations who have normal cognition. But strategies that are developed to reduce dementia prevalence that are based on the CADI are somewhat problematic because, of course, it contains risk factors that we can't do anything about. So age, genetics, things that we can't change. And these risk scores might actually mask how the different risk factors within them are actually affecting clinical outcomes. And this is something that I've become really interested in testing. Okay, so this is work from a recent 2022 paper of ours that I just wanted to talk you through briefly. So in this paper, we wanted to determine how 2 different versions of the CATIE risk score were actually associated with hippocampal atrophy. So you can see there's a picture of a hippocampus. It's Latin from Greek, translating to seahorse, as well as episodic memory decline over a period of about 10 years. And we wanted to do this in people who were classified as amyloid negative and amyloid positive. So if you don't know, a buildup of amyloid plaque in the brain is a pathological hallmark of Alzheimer's disease, and amyloid accumulates over time very slowly. So positive for amyloid in this study just means that the level of amyloid in the brain was high enough to be reaching some type of threshold for it to be classified as abnormal, whereas amyloid negative meant that the level of amyloid in the brain didn't quite meet that same threshold. And this is data from about 600 people from the Australian Imaging Biomarkers and Lifestyle Study, which is a really large study that's been running for over a decade here now in Australia. And all participants were cognitively normal at the time of entry into the study. So we had 2 different versions of the CATIE. One of them included both the non-modifiable and modifiable risk factors. The other one included only the modifiable risk factors. And higher scores on both of these measures was indicative of greater dementia risk. Okay, so in terms of what we actually found, we found that when we used the version of the CATIE risk score that included both the non-modifiable and non-modifiable risk factors within it, in people who were classified as amyloid positive, this higher CATIE risk score was associated with a greater loss of hippocampal volume over time. But what our data revealed was that because those risk factors were all smushed into the one score, we found that this effect was likely actually only driven by age and carrying that E4 gene. So those factors are really robustly associated with amyloid accumulation, and so they were actually driving this effect. So to separate and tease that effect out a little bit, we then used the modifiable CATIE. And what we found was really interesting. So in people who were classified as amyloid negative, so they didn't reach that threshold for abnormality, a higher modifiable CATIE risk score was negatively associated with greater decline in episodic memory decline over time. We then wanted to tease that apart even further and look at the individual modifiable risk factors. And when we did this, we found that only higher body mass index was associated with episodic memory decline over time in people who are classified as amyloid negative. So to summarize what this all means, we found that having fewer heart health risk factors was associated with less memory decline over a period of about 10 years. So that's about a decade, but only in people who were amyloid negative. And when we pulled those risk factors apart, we found that only higher body mass index, which is a known heart health risk factor, was associated with that memory decline. And this suggests that in the absence of those abnormal pathological processes in the brain, that early intervention to actually kind of reduce these heart health risk factors might be beneficial in terms of reducing people's risk of developing dementia. And I know we've talked a lot about midlife modification of heart health risk factors, but I really want to stress here that it's never too late to be making small changes to your lifestyle. I've put a figure in here from the Australian Heart Foundation that I've just modified a little bit, which says protect your heart. But what we know now is that they also protect your brain. So to stress the message, get moving, reduce your stress, drink less alcohol, and most importantly, if it's been a while, go to the GP and get a heart health checkup. So thank you very much.
Thank you, Emily. All right. So I'm just going to, after all of that, all of the talks tonight, I hope you appreciate that there's a great importance around modifiable risk factors. It's never too late. We can start making changes. And so, what I want to talk to you now is a trial that we are running. As we know, multi-domain prevention intervention trials investigate whether modifying risk factors can impact thinking and memory and dementia risk. There are multiple of these currently ongoing and have already been conducted. However, a lot of these trials have been conducted only in older adults. And now the results of some of these trials do suggest that modifying these modifiable risk factors, so targeting heart health, targeting sleep, targeting social isolation, do work, and they do help prevent cognitive decline. But again, most of these trials have been conducted only in older adults. A lot of them are quite inaccessible in the sense that everyone gets given a very prescribed intervention, and more importantly, it is not personalized to the individual specific circumstances, right? So we understand that all of these modifiable risk factors are largely interrelated, pretty, you know, as Lisa and Amy have alluded to earlier, you know, having poor sleep is typically also related to poor heart health. Poor heart health is typically also related to poor mood. And it is often that you find clusters of these modifiable risk factors that happen together, and they very, very rarely sort of happen in isolation. And so what we want to try and do is see if we can target some of these modifiable risk factors to prevent cognitive decline. So what is BetterBrains? BetterBrains is a person-centered 24-month fully online lifestyle prevention trial. What we've done is identified 4 modifiable risk factor domains. So 4 of the main ones. This is poor mood, so you know, depression, anxiety, depressive or anxiety symptoms, stress, poor sleep. So any kind of reports of symptoms of insomnia, any potential hints of obstructive sleep apnea, but also any sort of changes in circadian rhythms that you might notice. Poor heart health, so low physical activity, poor diet, but also things like hypertension, hypercholesterolemia, and then also low social and cognitive engagement. So this means social isolation, you know, kind of low cognitive engagement, things like that. And what we want to do is target these 4 broad domains. So when you come into the trial, you will sit down with a clinician who will take you through, will put you through a whole bunch of assessments, a lot of different questions to assess your current status. Obviously, if you don't have any modifiable risk factors, then that's great. We say, well done, you're doing all the right things. There's nothing for us to do here. But if we do identify any modifiable risk factors, then you will be randomized. So like tossing a coin, randomized into whether you receive the intervention, which is this personalized intervention that I'll be talking to you a little bit about, or into the control group. Unfortunately, we won't be, because it is a trial, we can't give every single person the intervention. Hopefully, if we find that the trial does work and the intervention does work, we'll be able to roll this out more broadly into the population. But at the moment, what we're testing is our hypothesis that intervening in these 4 broad domains of modifiable risk factors will be able to slow memory and thinking decline in people who are at risk of developing dementia. And by at risk, we are defining that as people who have a family history of dementia. So we are actively recruiting. We are hoping to reach about 1,500 participants. You'll see that we're about 75% enrolled. We've randomized about 40% of people. So there's still time to join the trial if you're interested. And of course, if you have any questions about the trial in more detail, you can always email us at betterbrains@monash.edu. The study process is that you'll go through an interactive consent process. You'll read the explanatory statement, create an online profile. You'll have to do a risk factor assessment, and this is where we ask you all sorts of questions about your mood, about your sleep, all sorts of things like that, and then we determine whether you actually have any risk factors for dementia. And if you do, you'll need to do a baseline assessment, and then from there you'll get randomized into intervention or control. The journey, if you do get randomized into the intervention group, you will be, as I said, assigned to a clinician, and this person will work with you. And this person is typically either a dietitian, a psychologist, a physiotherapist, who will work with you and look at your profile of risk factors and target the ones that are relevant to you. It will be a co-design process in the sense that you will be asked to identify goals that are attainable to you. So for example, if you are somebody with somebody who doesn't exercise, feeling a little bit depressed, but in the conversation with your clinician, you identify that, okay, I'm not ready to sort of see a psychologist at the moment, but I'm ready. I can go out and sort of take some steps to increase my physical activity. Those are the goals that you will then set. And the idea here is that we want to meet people where they are at, right? So a lot of the times, a lot of these things, what to do. I think that most people know that we should sleep, get enough sleep, eat well, exercise well, all sorts of things like that, but we don't actually do them. And so, having this support system, we're hoping will be a bit of a boost to that motivation. Some of the other features of the trial is that it is fully online, so anyone can sign up. In fact, we have about 40% of our participants are from rural or regional Australia, which we're really excited and happy about. We do sort of these monthly and weekly sort of check-ins to make sure that you keep on top of your goals and monitor your progress. We do have a smartphone app that sort of tries and makes that process a little bit more seamless. And then in the control group, or in the control and the intervention group, both groups will get access to what we call brain blogs. And these are just these educational blog posts that are specifically written and curated by our expert investigator team for our participants, and they usually cover some relevant topic relating to dementia. So this is the eligibility criteria if you're aged between 40 and 70 years old inclusive. So if you are turning 70 in 2 weeks time, we would still love to have you. Similarly, if you're turning 40 in 2 weeks time, we're happy to wait for you to turn 40 before enrolling you into the study. If you have a family history of dementia, so it doesn't sort of matter if you don't know 100%. This is really a self-report measure. If you think that you have some kind of family history, any family history really, it can be your mom, dad, aunt, uncle, cousin, grandmother, grandfather. We're looking for anyone with a family history of dementia. If you have a laptop or desktop computer, because you'll need them to do the testing. And obviously, if you want to make changes to your lifestyle, please do let us know. We would love to have you be a part of our trial. So this is our website, betterbrains.org.au. We have a little video for you to watch on the website as well. And if you have any questions, please do email us at betterbrains@monash.edu. So I think with that, we have a lot of questions as a Q&A panel. I'm just going to stop sharing so you can see all of our panelists. And can I ask all of our panelists to turn their video on? I'm going to try and curate some of these, and I might actually direct some of these questions to Amy first, because I know Dr. Brotman is very, very busy and has another Zoom meeting to go to after this. So I might just try and get some questions for you first, Amy.
I can comment on a couple while you're doing that.
Yeah.
So the first, I noticed there's lots of questions on how to get your ApoE4 done. You can. It's not covered by Medicare, so you have to pay for it. It's not super expensive, but your GP can order it. There's lots of labs around Australia that will do it. Just note that some people are concerned because they feel it might have life insurance issues. So far in Australia, that hasn't been the case, but you can get it ordered anywhere you like, but you will have to pay.
Yep. And I'd just like to add to that too about ApoE. A lot of doctors don't encourage people getting tested for ApoE just because there's not really sort of anything that we can do about it. It is not 100% predictive. So just because you have the E4 allele doesn't mean that you're going to get dementia. And similarly, just because you don't have the E4 allele doesn't mean that you won't get dementia either, right? So it's actually, it's pretty, it actually does a pretty bad job at predicting who will, at an individual level, who will and will not develop dementia. And so primarily that's why a lot of doctors discourage you from getting tested for it. So we have a couple of questions about hearing loss, Amy, that I thought you might be able to answer. So is neurological single-sided deafness a hearing loss risk factor? As in, I had a virus in the inner ear and I'm deaf in one ear.
So I think that that's probably not what we'd regard as sort of the age-related memory loss, age-related hearing loss that's common. But we think that any hearing loss, and a lot of people have got industrial hearing loss as well, particularly veterans have got noise exposure. We think it's probably related to the fact that they're missing that social discourse, the fact that the interaction that we have is very incidental. When we look at risks of depression in people who lose their sight versus lose their hearing, it's much higher in people who lose their hearing, because we need that sort of, our auditory cortex, that part of the brain for hearing, sort of is switched on all the time, and it's a very important part of monitoring our social environment. So we think that hearing loss, whatever the cause, is potentially associated with a risk. One of the questions was, is it because of the dementia? No, it's not because of the dementia. It's not, there's no cause of dementia that I know of that actually causes hearing loss as well. There's lots of neurological causes, but that they don't cause dementia, they're quite separate. And there was one more question, which was whether congenital hearing loss is associated with an increased risk of dementia. It's not known. I haven't seen any good studies that have looked at this. The fact is, if you have congenital hearing loss, you often learn, well, nowadays you often get a cochlear implant, but you used to learn how to sign, and so you became embedded in the deaf community. And that in itself is very protective, because that's very social, very protective. So we don't believe that congenital hearing loss is associated with dementia later in life.
Excellent. Thank you, Amy. And I also have one extra question for you. This is from Jenny. My maternal grandmother had dementia, type unknown, and lived to 102. My mother was diagnosed with vascular dementia at age 82. So about the same age, my grandmother showed signs of dementia and has no other health issues. So medical people have indicated that I'm also likely to get dementia. I'm 58 and female with a current BMI of 28. It was 43 18 months ago. Good on you, Jenny. Is it true that I am likely to inherit dementia, or is this only the case with early onset dementia?
Yeah, I think none of these are the things that I'd be concerned about, Jenny. I don't think 102 is remarkable, and 82 with some vascular changes is also remarkable. You've inherited potentially a genetic risk for vascular risk factors, so you probably inherited a risk for stroke and heart disease, but I don't think you'd be worried about your dementia risk from that history. And we know that people, there is now evidence that people who inherit that sort of general genetic risk, which might be associated with the ApoE4, in fact have more to gain by having a neuroprotective lifestyle. Certainly from an exercise point of view, there's very good evidence that the most gains from exercise are from people who've got the ApoE4 allele.
Thank you. Now here's an interesting one. So should those at increased risk, so for example, they carry the ApoE4 allele, take prophylaxis statins from their 30s and their 40s?
Oh gosh, no. So no, we don't. It's a really good question. We know now. I'll give you a bit of a long answer. We used to think that perhaps for those most at risk that they should take aspirin. So that's aspirin, as many of you know, will thin your blood a little bit. However, there's now some beautiful big studies, in fact, done in Australia, the Esprit study, showing that for primary prevention, so that is starting it before you actually have any of the manifestations of the disease, For heart disease and for dementia and for stroke, primary prevention, aspirin doesn't work. It hasn't been done with the statins for a number of reasons. They're not probably as benign as aspirin. They do have side effects, but we know that the statins are associated with cholesterol, and that potentially cholesterol treatment is good for vascular health. So we're actually mediating the benefits to the brain via vascular health. There's no evidence for heart disease, at least, that statins work to help reduce your risk. And there's minimal evidence in stroke, and very, very little evidence in dementia as well. So no, I wouldn't be recommending that. I'd be recommending a Mediterranean diet with lots and lots of olive oil and some fish, and lots of vegetables, nuts, seeds, the things that we've spoken about.
Great advice. Okay, so a lot of questions actually about amyloid and amyloid scores. Paul, do you want to address that?
Sure. So a lot of the work that we talked about with amyloid in the different data that you saw on this, on the presentations, and indeed some from me, was determined from PET imaging, which is a nuclear medicine procedure whereby a dye is injected into the blood, attached to a radio tracer. The dye binds to amyloid particles in the brain, and the radio tracer accordingly does. And then we can detect that binding with a camera. At the moment, this is still a research tool, so we don't use it clinically as a diagnostic tool for dementia as yet. That being said, there's given the way in which medicine in Australia works, where we are assertively trying to identify and treat people with this early signs of dementia. If you're interested worried and interested, there's many avenues through signing up to different research programs on getting access to one of these scans, and then depending on what happens with the scan, maybe joining a research program. To some extent, the amyloid scanning is a lot like ApoE4. It's a sign. It's not indicative of the disease. It doesn't really give you strong predictions about what's going to happen in the next decade. There's those issues as well. So we're making broad population-based generalizations about our data. Of the extent to which many of these things work on an individual level, it still has a lot of variability associated with it. What will also happen with amyloid is that if we think tonight about the sorts of language that we've used in describing late life brain change, we've used words like cognition, and so we've said that's really thinking. And we take thinking as an external or an outside manifestation of how well a brain is working. And then we've sort of designed or decided that that might reflect two major types of insult, or thinking might change for two main reasons. One is there's changes to the way in which the blood flow in the brain operates. So that's, as we call, vascular changes, vascular dementia. And then there are changes in thinking that reflect the accumulation of these abnormal biological processes. One of those is called Alzheimer's disease. I noticed that there's been some questions about other dementias on the thing. Some people have asked about Lewy body dementia, Parkinson's disease. So there are other dementias, but as a proportion of dementias that occur, they're very, very low. And so the discussions that we've been having tonight are largely about Alzheimer's disease, which in these days is defined predominantly by the presence of the abnormal pathology. And that is what you determine from the amyloid PET scan, from the neuroimaging scan, and a cardiovascular disease or cerebral vascular disease that you often obtain or from a magnetic resonance imaging. Just while I'm on it, you can also determine amyloid levels from lumbar puncture, and there is now developing very quickly technologies that will allow us to detect it in blood. But these are all even more experimental than the imaging, and so still not as a general opportunity for people to come on. However, as I said, all of these are available through enrolment in research programs.
Yeah, thank you, Paul. And I think related to this is a question about whether there is a test currently available or in the horizon for dementia. And I think part of the challenge for that is that you can get a dementia diagnosis that involves going to see a GP and then getting a referral to a memory clinic. You'll be put through a couple of different tests, neuropsychological tests, cognitive tests. And your doctor will ask you a couple of questions about, and might also ask your caregiver, your partner, somebody who lives with you, a couple of questions about your ability to function on a day-to-day basis. However, I suspect that when person has asked about whether there's a test for dementia, I'm guessing you're referring to a blood test or a brain scan of some sorts. As Paul has mentioned, it is very, very experimental. We are conducting some studies where we are going to test whether certain blood tests will be useful and helpful in informing a diagnosis of dementia. But again, these are super, super experimental.
I think, Yen, it would be worthwhile clarifying too that I talked about how quickly medicine has been moving in this area, and doubly quick in the context of the term that we're using, dementia. Mm-hmm. So the way in which we sort of conceptualize this now is dementia is the clinical syndrome. Mm-hmm. And the major diagnostic criteria for dementia is that your thinking and behavioral changes have become so severe that you can no longer live independently. That's dementia. And then the next question comes as to what might be the cause of that dementia. And so you can have causes like Alzheimer's disease, you can have causes like Lewy body dementia, or you could have causes like vascular changes. So we have the clinical syndrome, dementia, and we have the causes, Alzheimer's disease. So it's only probably been 5 years since we called Alzheimer's disease was the clinical syndrome, right? It was called a. So when you're thinking and conceptualizing or reading about a test for dementia, reading about a test for dementia, think, am I talking about the test for the clinical symptoms, or am I talking about a test for the biology? And some of the arguments, and indeed the argument at the basis of the whole BetterBrains and modifiable risk factors are that the biology always precedes the clinical. And so if we can stop the biology occurring, or the bad biology, if you like, then ultimately you don't progress to a clinical syndrome of dementia. So that's the main idea.
Yeah, and we also know too that increasingly, very few people present with only one form of the disease, right? So Alzheimer's disease, even though it's the most common form of dementia, it often co-presents with other forms of dementia, like vascular dementia. I think it's only in very recent years that we've started to differentiate these different types of dementias. I think previously, particularly in clinical practice, the convention was always to diagnose somebody with the broad umbrella term of dementia, irrespective of what cause that is.
The reason being, of course, is that we are moving really quickly with treatments for different biological processes that we believe underscore dementia. So there's been already a number of drugs that have failed, admittedly, in Alzheimer's disease, seeking to stop that biology. But in order to optimize or to ensure that the right people get the treatment, understanding not just the clinical presentation, but the reason for that, the biology of that, is a crucial point.
Speaking of biology, is there a natural way to clear amyloid plaques in the brain?
That would be a good one for Shantha. I think it works.
Sorry, can you repeat the question again? I was reading.
I know you've been, Shantha, you've been very busy answering questions on the chat. The question is there a natural way to clear up amyloid plaques in the brain?
Well, so there is a strong body of evidence that sleep plays a critical role in in clearing a number of these proteins and waste products. So the increasing research that is showing that sleep, particularly deeper stages of sleep, have a role in the lymphatic system, which we know plays a role in clearing amyloid levels. So I think that is one of the ways in which we can actively reduce that risk factor.
Yep.
Thank you. Okay. Is napping as a supplement to night short sleeping beneficial, Shanta?
I was just answering that one, just got onto that. So napping, in general, more sleep is good. And as we age, a consolidated bout of sleep overnight is more difficult to achieve, right? So that's just the thing to keep in mind. I answered someone else's question to say that it's perfectly normal to have brief periods of awakening from sleep through the night. And as we age, those awakenings become more pronounced and it can be more difficult to fall asleep afterwards. Some people find that it's beneficial to try and supplement the nighttime sleep with a daytime nap. I was showing you earlier in my talk this thing called the homeostatic sleep process. What that means is that if you wait too long in the day and if you leave it till the afternoon and evening to have a nap, it means that when you wake up from the nap and by the time you're ready to go back to sleep again that night, there's not enough time that has passed. So your homeostatic sleep pressure hasn't accumulated enough for you to fall asleep again. So if you are going to have a nap, it's a good idea to have a nap very early in the day, as early as possible in the afternoon, so that you can build up that sleep pressure and still be able to fall asleep easily at night.
Thank you. All right, I think this is a question for Lisa. Have you come across if ethnic background influences the risk of developing dementia?
Short answer is yes. As I don't particularly research it myself, but I know from the literature that there is that Alzheimer's disease and dementia can manifest differently in different ethnic populations. So we know that particularly the Indigenous Australian population has a higher incidence and prevalence of dementia, and we see this across different groups as well. And we've seen that genes like ApoE4 also act differently across different groups, but it's very likely complex, related to genetics, and also maybe some groups are more disproportionately affected by certain modifiable risk factors. So it's definitely a complicated issue, and I'm sure the rest of the panel can agree that the field has probably only just scratched the surface of what needs to be researched to answer that question. But thank you.
Yeah, I just quickly want to add to that as well. I think this is an area of cognitive aging and dementia research that we really need to start building some momentum in. So we really need more work done in culturally and linguistically diverse populations. We also need better work to actually even just estimate the prevalence and incidence of dementia in these populations as well. So we know that Indigenous Australians in Australia have extraordinarily high rates of both cardiovascular disease and stroke, but also dementia as well. And this has only kind of just started to emerge in the literature. So there's a lot of work that really needs to be done here. And I did also just want to comment on a question I saw in the chat, which related to social isolation. So there's been some really interesting work over the past few years with COVID, and we've shifted everything online, which is related to life space. So, how often you actually exist outside of the confines of the same four walls of your house. And it's shown that, the more socially engaged you are, the more you're, meeting new people, connecting with community groups, getting involved in your community, looking after your grandkids, just getting out there, that is actually really protective for brain health. So just to encourage everyone, to pick an activity that you enjoy, research it, and get onto it. Just do it and increase your life space.
Thank you, Emily. I think there are also a couple of questions here about sex and women being at higher risk of developing dementia. Paul, do you want to answer that?
Sure thing. So the actual role of sex as a risk factor for dementia is still unclear. There's some populationistics. Come back to one of the points that I raised, is that oftentimes estimates of what we have about a risk factor for a disease were based on either biological changes or social changes that occurred when people were in midlife. So at the turn of the century, at the 2000s, when this issue of sex was noted, or even prior to that, it was certainly the case that men were dying younger than women. So women just lived longer, and because a major risk factor for brain disease is age, there were more older women than older men, and so there was more women with older brain disease. Mm-hmm. So that was where we started with this, and it's taken a while. Men are catching up now in their life expectancy. So this is becoming closer, and we're now starting to ask, are there biological reasons for the difference? There's no clear, strong evidence. So while these risk factor studies, the data that we've shown you are risk factors, and what they show you is correlation. And you'll hear us scientists boring everybody all the time with statements like, correlation is not causation. That means just because it's associated, it doesn't mean it causes. And so we have to ask, so we saw things like weight, increased BMI, increased cholesterol level, mm-hmm. Increased hypertension. We know in heart health that these things are under-recognized in women as well, right? So it might be that the female sex is associated with just different exposure to those other risk factors, and that's bubbled through into our estimates. That's one side of the issue. The second side of the issue is that there are actually aspects of biology about being a woman that will somehow either exacerbate the biological processes that are at the root of this disease. One of the big ones, of course, is sex hormones, estrogen. And mm-hmm. while there are still data, so there's some data suggesting that women who go on to postmenopausal hormonal therapy early do better in terms of reducing their risk of dementia later on. Again, the size of those effects are relatively small. And so whilst they might be interesting experimentally, the extent to which you would guide an individual's behavior about might be a bit limited. And then other things like hormonal changes and risk for other diseases that occur more commonly in women. So the jury's out in terms of, does female sex cause dementia? But the epidemiological or statistical evidence does suggest at this stage that there's something about being a woman in the 21st century, which probably reflects what happened to you in the 20th century, that does increase your risk slightly of Alzheimer's disease.
Yeah, it's an interesting one, isn't it, Paul? Because male sex is typically associated with greater cardiovascular risk. And as we've heard over and over again tonight, cardiovascular risk factors are some of the key risk factors for increasing dementia risk as well. So it is one of those things that is I think can be quite difficult to tease out. I think there's some emerging data that suggests that perhaps this menopausal period might be really key to determining whether a woman is at increased risk of developing dementia or not. But like you say, the jury definitely is still out on this.
Oh yeah, and so so we come back to Shantha's work. I mean, one of the great manifestations of the menopausal period is sleep disturbance, right? So so there's all sorts of pathways by which these things, but so it's a factor that we have to consider. It's certainly a factor you can't modify, although that's becoming less the case these days too, right? And so it's something it's an interesting and important clue at this stage, I would say.
Yeah. Thank you. All right, so we have come to the end of our session. I'd like to thank all of our panelists. Amy had to leave early, but thank you, Paul and Shantha and Lisa and Emily, for hanging around and answering some of these questions. I know that we weren't able to get to every single one of them, but thank you everyone for dialing in, for tuning in, and spending your Thursday evening with us. If you have any questions, please email us at betterbrains@monash.edu, including questions about how to get involved in some of these studies. We are also really active on social media, so if you want, you can please do follow us at healthybrain_AU on Twitter. We're on Facebook, and of course, you can also log on to our website as well. So thank you very much, and have a very lovely evening.
Thank you, everyone.
Thank you, everyone.
Thanks. Bye.